Thank you. Thank you to all the speakers for their excellent presentation. We could all agree that there is a clear understanding of the intricate relationship between the virus and the host, and it's so imperative to gain this knowledge in order to develop more effective vaccines or therapeutics. We now could open the stage for questions. Teresa, do you have anything else to add at this point?
No, simply that if we cannot go over all of the questions because of lack of time, please feel free to reach the speakers and, you know, in gather time, if you can actually, your avatar can catch them when they are moving around. So, no, without further comments, again, I also thank greatly all of the speakers. I think they were very interesting and excellent talks. And we'll start with some questions, I guess. Elizabeth, your
yes,
turn.
I believe there is a general question that could be answered or stimulate the discussion among all the presenters today. And the question is, if an attenuated FNDV is to be approved, how important for the regulators is to differentiate these biases? And when we say biases, we are talking about codon bias, CPG, UPA, and or RNA structures. So what the presenters could comment on this question?
So I can probably provide some answers, but we still don't have the right answer. We have not done experiments that can differentiate between codon per optimization and the CPG dinucleotide frequencies. I mean, these are changes that happen unintentionally when the genome is recoded. However, if the question is referred to safety, we have conducted passages of these viruses in cells up to 15 times. And regardless of the unintentional increase of CPG dinucleotide frequencies, we see that the viruses are genetically stable. And so we feel that there probably won't be a gain of function if that's a concern for the regulators that are involved in approving these vaccines. But of course, we will need to establish appropriate safety testing to prove our hypothesis.
I would add to Giselle's comment that also the stability of those genomes and the distributions and the fact that you probably need a marker also to label these genomes could be parameters that could be also be considered by those regulators beside what you just said. Reversion to virulence is a critical characteristic that could be considered regardless of the modification that has been introduced. Any other comments on the topic? If not, I would move for a question for Ian. And I think this requires more a clarification of some of the statement that you did. And this question is as follows. If understood correctly, you are indicating that there is no fitness benefit for the recombinant virus. That's the question. But could it be possible in the future study to tweak your model system to explore between pairs of viruses where the recombination has a clear advantage?
Yeah, so in vitro, it didn't look like the recombinant, or at least the one that we were looking at specifically, had any fitness advantage in cell culture growth. But in vivo, in the animals, we found these recombinant viruses were usually dominant for this window of time. So I think they probably were. I did have a fitness advantage in the animal during that window when, they were able to evade neutralization, that kind of thing. But yeah, I mean, if we did a co-infection experiment like this where we had, where we used mutants that, somewhere far 3' there was something bad going on one of them and vice versa, I think we'd find that, whatever recombinant came out of there, if it was more fit than its parents, then it would probably succeed. I think some of the future of these de-optimized viruses could show something like that too, if there is any chance of recombination with those. I don't know if there is really, but yeah, hope that answers the question.
Okay, thank you, Ian. Then we have a question for Caroline, right? And you mentioned the difference effect of the mutation on RNA structures in primary interferon competent versus immortalized cell lines, presumably to the interferon competency. Have you evaluated your constructs in different immortalized cell lines to see if that holds the same phenotypes?
I think, yes, we have looked in BHKs as well. And again, we didn't see the attenuated phenotype of the RNA secondary minimized virus in BHKs as we didn't in the ZZR cell line. I'm trying to think if we've done it in anything else. No, I think it's just those two in terms of the sort of cell line rather than the primary cells. Yeah.
Thank you. Teresa?
I'm trying to. So I guess for Margarita. So a question from the audience is, can you, Jean, edit the RLRs to test whether they are resistant to the LPRO activity?
Yes, that's actually a very nice, very interesting point. Yeah, I guess you could do that. Actually, we were planning on doing the knockout of MD5, but COVID-19 came and the lockdown, and so we couldn't do it. But similarly, that people, you can do knockout with CRISPR or whatever. I'm pretty sure it wouldn't be that difficult to generate a cell line with cleavage-resistant MD5 and test how their susceptibility to viral infection looks like. So yeah, it's definitely a very interesting point.
And sort of related, have you found that the target sites on the RLRs like MD5 and LGP2 by the leader protease of FMD are conserved across species? So the target recognition sequence?
Yes, they are, absolutely, because it's located in a critical motif for activity of the RNA helicase. So yeah, actually we have checked the human sequence, swine sequence obviously, and bovine sequences. And yeah, they are absolutely conserved. Yeah, not for example, as you, I don't know if you noticed in the presentation, between them, there are slight differences for the regi, the flanking residues are slightly different, but we are not sure if that contributes to the fact that we don't detect cleavage on regi. But in the case of MD5 or LGBTQ, it's absolutely conserved among species. Yeah.
So, so thank you, Margarita. So we'll try to go around, and if then we have time, we have more questions for you that are posted on the chat, but also maybe some others will come up after this discussion. So, so I have some questions for Lydia, I believe. And so let me see if we see anybody in the audience that may have asked questions. So one question I have is when you mention, and perhaps I miss it, that for this study and to determine whether RNA structures were in part important in the genome, in the ORF for fundamental FMDB activities, you mentioned that you align, you apply your software to the alignment of 118, I believe, FMDB sequences. So, and then you mentioned that the identified sequences were conserved across serotypes. But in your alignment, how diverse, or how was the representation of the 7 different serotypes considered, let's say? So do you think that there would be a bias to some particular serotypes based on the analysis?
So we try to minimize non-biased, and we approach it by downloading literally everything what is the full genome sequence at the GenBank. Also, we perform some sequences because, as everyone in here is aware, the SAD viruses are greatly unrepresented in terms of the genome availability. So we also sequenced some of the SAD viruses, which we are very lucky to hold as a part of the FMDV reference laboratory. And we designed the study in such a way that having all those full genome sequences, we performed a phylogenetic tree using VP1 as the most variable genomic region. And then we make sure that we have a representation for each cluster. So actually the data set in here should represent the genomic variability of FMDV known currently. And we tried also to make sure that there is no, so for instance, you don't have 50 of O's and only 3 C's or SADs, so that the proportional representation to different serotypes isn't the same kind of number. So one serotype shouldn't enhance structure prediction against the other serotype, if you know what I mean.
Mm-hmm.
And I should probably mention, so one of the things which is very tricky when you perform the full genome alignment will be the fact that the capsid region is so variable, so you can't perform the alignment in that region very, very well. But we use an alignment which doesn't consider only a nucleotide identity, but also RNA structure. There's a MAFT-specific alignment when you can align structures. You can align genomes not only by nucleotide identity, but actually it considers predicted RNA structure in there as well.
Okay, thank you. So I don't know, Elizabeth, do we have?
Yes, I'll continue with a question presented by the audience to Giselle. And the question is the following: does infection of cells with codon de-optimized viruses result in the secretion of greater levels of type 1 interference? In other words, induction of enhanced innate immunity.
So in the studies that we have conducted with the codon per de-optimized virus in the P1 region, we haven't analyzed that data yet in cells because the viruses are so attenuated that we will need a cell line that not necessarily a primary cell line, but a cell that has an intact innate immune signaling response, and that has become a little bit of a challenge. However, we have evaluated the mRNA transcripts from PBMCs that came from pigs inoculated with the de-optimized viruses, and we have examined mRNA transcripts that encode proteins involved in antiviral responses or other innate immunity signaling pathways, and we have not seen any significant upregulation. So we don't think that the de-optimization in these viruses are enhancing any type 1 interferon signaling pathway or other antiviral response.
Thank you, Giselle. Ian, I think we need to go back to you with the clarifications. I think a couple of members of the audience posted some questions regarding if you could elaborate more what you mean by the success of novel viruses over the A viruses due to the immune evasions. I think that concept is being requested for further clarification at your side.
I think we're out of time. I'm seeing our
yeah.
Yeah, so something's about to start. So, yeah.
Yeah, perhaps Ian, you can save your answer for the Gather Town, or
okay.
Yeah, and yeah, we should close the session now. Everybody's saying we have a session in 5 minutes. We want to thank very much all of the speakers on this session for the excellent work, and okay, we'll meet you later during the meeting. Enjoy the rest of the meeting. I don't know if Elizabeth wants to say something.
Thank you, everybody, and we'll continue with these interesting discussions later on the chat or at the room, Gather room. Thank you.
Bye.